Well, thank you very much to all of you to be here. Well, I would like to start to ask you a simple question of how many of you think you are who you are because of what is written in your DNA. And, you know, this is a question that I asked to myself since a long time, maybe when I was a kid. And this is maybe the reason why I decided to start studying biology and genetics. And I have to say that for many years, scientists really believed that by knowing what is written in your DNA, we could help to predict your future, who you are, and what type of disease you're going to inherit in your life. However, today we know that this is not necessarily true. I mean, the DNA alone cannot really fully explain who you are and what type of traits and disease you will develop in your life. And for this reason, we are looking right now at other type of molecules that can work along with the DNA to transmit information from one generation to another. So this new way of transmitting information we called epigenetics, and is something that deals with the DNA and with the information related to it. So you might maybe know that the DNA in all the cells of your body is actually highly organized and is wrapped around specific structure that is like balls. And those are actually, it's very important because those structures can be heavily modified by many chemical modifications. And we call this modification epigenetic modification because are really the substrate for transmitting this memory. Now, what is important is that these modifications are in the proximity of the DNA, and they can help to reveal what type of information you have in your DNA. Now, there is one important feature you need to keep in mind is that this epigenetic modification, they can change during the course of your life in all the cells of your body, especially when you are exposed to environmental perturbations such as what you eat or even what type of exposure to pathogen or toxin you have in your life or even the aging. All these things can lead to changes in this epigenetic modification that are in the proximity of your DNA. Then the second important point is that those epigenetic modifications can also be inherited. And so you can imagine how your lifestyle or what you experience, let's say exposure to starvation or high-fat diet, or even traumatic stresses or toxin, they don't only have an influence directly on your life, but they can also perturbate and affect the life of your future generation. And for example, these epigenetic modifications that are inherited, they perhaps can contribute to the development of metabolic or neurodevelopmental disorder. So you can appreciate how it is important to now understand more and more how this epigenetic mechanism works to understand and predict maybe disease heritability in human population. I have to tell you that studying this process in humans is extremely difficult. And first of all, because we are all different. We are all genetically different, and so whenever we want to trace, something about our diseases, we never know if it's because we are genetically different or there are other epigenetic molecules that we inherit. And then the generation time in humans is extremely long for an experimental biologist like me. If we want to follow multiple generations, this will take hundreds of years, and I don't think I have this time. So we had to come up with let's say, a simplified version of human beings, and I think those are worms, and in fact, I do think we are like worms. So this is a very special worm that we use in the lab. It's called C. elegans, and there are two very important features of why we use it for epigenetic research. So first of all the worms are all genetically identical. They are all like twins. And so you know that nothing is changing in their genetic, and therefore we can really study very well what epigenetic changes can lead to in this animal. And then second, the generation time is very short. So you see it's like 3 days, so basically you can follow multiple generations in a couple of weeks, and this is useful for us that we do experiment in the lab. So I told you that these worms are all genetically identical, and this is because these worms are hermaphrodite, meaning they have both sexes, and so they can make baby alone. They don't need to do sex. And they do actually a lot of babies. So here on this graph, you see each dots correspond to the progeny of one individual worm. So basically, every worm makes like 200-300 babies. So this is quite a lot. So I was interested in this fertility phenotype in C. elegans, and we were studying in the lab at the beginning of my lab, like genes that are involved in fertility. So now, if you have a gene that is essential for fertility, usually if you mutate it or you delete it, basically you expect that the worms are all sterile because the genes are not working anymore. But when we did this experiment in the lab, surprisingly, the mutant worm, they are all inherited this mutation, but they were not sterile at all. Actually, they were making a lot of babies, like 200-300, like the normal population. However, if you now follow this baby across multiple generations, now we start to observe a gradual decline. And you see these are generation 7, 8, 9, up to 12 generations. All the worms are now finally sterile. There are no babies. They cannot make babies anymore. So this is now very interesting because these worms are all identical. They have all the mutation in the gene that we thought that was causing sterility, but they acquired the sterility only later on. And because of this feature of the worm, we were able to understand what is causing the sterility. And in fact, we discovered that epigenetic molecules, the one I described earlier, these are the ones that are inherited together with the mutation, and they are causing now this sterility. So with this work, what we let us understand is really how diseases might be erased in a population, which is not only caused by the inheritance of genetic mutation, but it is also a contribution of all these epigenetic molecules that are inherited together with genetics, and they lead us to discover diseases, okay? So now with this story of the world, you might think that epigenetic inheritance is all bad. I mean, you know, we inherit these molecules, we get diseases, and that's it. But I want to tell you another little short story of worms where I can show you how instead these molecules can also be helpful. For example, memorize life experience that can be useful for the future generation. So this is a study that has been done in a laboratory of a colleague of ours in Princeton. And so they are studying basically how the worms choose among different food. So the worms eat bacteria, that's what they like. And basically, if you let these worms choosing between different food, like let's say a healthy food and a poisoning food, you will be surprised, but the worm is attracted by the poisoning food. They all go to the poisoning food. However, after they eat this food, they get sick because it's poisoning, they colonize the intestine. And so if after 12 hours of exposure, you then let them choose again what type of food they will like, you will see that now they learned to avoid the bad food, they really dislike it, and they go to the healthy food. So this is a learning behavior that they acquire upon an experience that they have, because they experience the poisoning food. Now, what is very interesting is that if you take the babies of this worm and you let them choosing between the two food, and they never seen this food before, so normally they should go to the poisoning food, you will see that now these babies, they go to the healthy food, which means that they inherit the memory of the experience that the mother had in the previous life, okay? And this inheritance can last for 4 generations. So the memory of this experience can be pretty long. Now, we know what type of molecules that might be involved in this process. As I said, is epigenetics. But the central question that we are investigating in the lab right now, in this current research at the moment, is to understand how now these epigenetic molecules that are changing in the tissue or in the organ that is directly exposed to the perturbation. Let's say the brain, when you are experiencing some experiences in life, or the gut when you get colonized by the bacteria. So all these epigenetic molecules that are changing in those organs, at some point they need to be inherited. And so the central question is how this is possible, because it means that from this site of the intestine or the brain, these molecules need to somehow to move in the tissue that is the gonad that give rise to the next generation. And this is now a very important question. And we think we know the molecules that can travel across tissue and that can bring this memory. And this is a new molecule that is very interesting in the epigenetic field. It's called small RNAs. Those are really little pieces of RNAs. And RNAs is a molecule, it's similar to DNA, but it doesn't matter. But now those molecules can be central to travel and to transmit information across organ and to go to the next generation. And in the lab, we are now working on specific small RNAs that, for example, are produced in the intestine, are delivered to the baby, and this is very important because these baby now that acquire these molecules, they are better surviving different stresses in future generation, okay? So I hope I convinced you that with the story of the worms, epigenetic inheritance is not only deleterious, meaning it's not only allowed to inherit diseases and so on, although it's important to understand this, but it can also be important to transmit environmental experience that we live in our life to maybe better prepare future generation to future challenges. And with this, I just would like to thank the people. This is obviously, you know, when we work in the lab, we are a team of scientists, the Institute Pasteur, and you are all welcome to visit the lab, Pasteur, and especially the worm. So thank you very much.